Okay, are we ready, everyone? Yes, we came back to Oakland as we promised. So good evening, everyone. Welcome to Science at the Theater's 5 Big Questions, brought to you by the Friends of Berkeley Lab. My name is Jeff Miller, Head of Public Affairs at the lab, and I will be your host for this evening. Now, we have something special planned. We're going to have a voting segment at the close of the 5 scientific presentations. Each will be 10 minutes each. And you can't see it, but we have a timer up here to make sure that that's the case. And there'll be a beeper at 10 minutes. We're going to ask you to consider, of all the questions you've heard, which is the one that you would like science to answer first. Okay? So you're going to be able to text to vote, or for those of you who prefer a paper ballot, you can do that as well. We'll hand them out and collect them and tabulate them with the help of our volunteers. Now, there is no prize, sadly. We are a government institution. But we think that the format will sort of stoke the fires, competitive fires of our scientist presenters and make for a very lively presentation. We hope you agree. Okay. So now to get the evening started, we are going to show our new Berkeley Lab video, which is short by design, a mere minute and 45 seconds. We hope that you enjoy it, and then I'll be back at the close of that to get the evening officially started. So please watch and listen and enjoy. Thank you.
Is it possible to make fuels from sunlight? Can we stop breast cancer in its tracks? Save consumers billions of dollars in energy costs? At Berkeley Lab, finding answers to the toughest questions is why we come to work every day. Our science paved the way for medical imaging devices like MRI machines and PET scanners. We added 16 elements on the periodic table, helped develop energy-saving windows, discovered the accelerating universe, and won 13 Nobel Prizes. Scientists from around the globe use our powerful supercomputers to do everything from develop new materials to predict the Earth's climate. We're working to create affordable batteries that enable electric vehicles to go 300 miles on a single charge. Berkeley Lab scientists have even created a solar-powered vaccine refrigerator that could help save countless lives in the developing world. Public service has been part of our DNA for more than 80 years. As a Department of Energy national laboratory, our science is open, ingenious, and humanitarian. It is done for you and belongs to you. The challenges now facing us in energy, health, and the environment won't wait. Neither will we. Our science, your science, will lead us to solutions we can't even imagine. Berkeley Lab, bringing science solutions to the world.
So is there anything that you noticed about that video other than the wonderful narration by Peter Coyote? Did you notice that it started with questions? So can we make fuel from sunlight? Can we stop breast cancer in its tracks? This was not done by chance. Questions drive science, and questions that provoke other questions keep science fresh and alive. In fact, if you think about the root of the word question, quest, it's really about a journey. It's about momentum. It's about a zest for answers that scientists have and need to keep propelling their research forward. So what are the objectives of all these questions? Well, sometimes it's just simple understanding. Sometimes it's a new technology. Sometimes it's just pure curiosity. But the point is that the questions never stop, and neither do we. At Berkeley Lab, questions are the brain food that fuel our discoveries that we think are in the public interest. Now, sometimes the questions seem a little esoteric. I mean, you might have looked at the program tonight and said, what is that? What is that? What's the relevance of that? Well, it's up to us to convince you otherwise. Thankfully, that's not going to be my task tonight. That's going to be up to our scientist presenters, who include Judy Campisi, who's going to be talking about age-related diseases, Kai Vetter, who's going to be talking about radiation, Javier Navarro talking about insects and why we should care, and we really should. And Almgren is going to be kind of a math thing, but math like you haven't heard it before, which you really like. Trust me, we know this to be true. And last, Shashi Buluswar, who will be talking about sustainable urban food production and the consequences for a city like Oakland. So with that, please give a warm welcome to our first presenter, Judy Campisi, who is going to be answering the question, what causes age-related disease?
Oh no, I'm talking about nuclear energy.
No, you are. There we go. Please.
Thank you. Thank you. Welcome, everyone. So let me start with a question to the audience. How many of you would like to live to be a ripe old age? Oh, good. Okay. So this is what you have to look forward to. So this is obviously a partial list of the many, many diseases that afflict us as we get older. And we are always surprised when you think about these diseases, something that affects your brain, something that affects your brain, something that affects your bones, your eyes. All of these diseases have one thing in common. Very rare in young people, and then about halfway through our lifespan, so 50 or 60 years in the case of humans, all of these diseases rise with very, very, very sharp kinetics. So there are two possibilities. One is that this is bad luck. All these different diseases, different tissues, different manifestations, just by chance, happen to come up at the same time. But the other possibility is that it's not a coincidence, that there are basic aging processes. There's something about aging that's driving these diseases. Now, if we're right, think about it. This will revolutionize modern medicine, because now there won't be a cardiologist looking at your heart and an eye doctor looking at your eyes and a bone doctor looking at your bones. Now we will be training doctors to treat aging, and that will simultaneously treat many of these diseases. So this is a dream. We're not there yet, but this is a dream. And what I'm going to tell you about today is the progress that we've made, a little bit of progress, in identifying these basic aging processes and then hopefully developing interventions. By the way, we can do it in mice. We can't do it in people yet, but we're getting there. So how do you go about identifying something that can drive all these very, very different diseases? And I'll show you one example of how we did it. So we looked at all these diseases, and we realized something striking. Most of these diseases are degenerative, and by that I mean things don't work very well. So your eyesight doesn't work very well, your bones get frail, your muscles get frail. But there's one disease that follows the same trajectory, rare in young people, comes up at about the midpoint of the lifespan with very sharp kinetics, that's different. And that's this disease here, cancer. It's very difficult, would be very difficult for me to call cancer a degenerative disease. In order for a cancer cell to form a tumor that will cause lethality, that will kill you, that cancer cell has to acquire new functions. So think about it. It's like most of these diseases are loss of function diseases, and then there's this one disease which we call a gain of function disease. It has to acquire something new. And we thought this is a good thing because what it can do is simplify the question. Now we can ask, what does cancer have in common with an aging brain or an aging bone or an aging muscle? So we found an answer, or at least a partial answer. And that answer comes from knowing what we know about cancer due to many, many decades of cancer research. We know now that there are two things that are needed to form a tumor that is lethal. So one is mutations. These are not mutations you were born with. These are mutations that you acquire as an adult or even as a young person, and those are cells within your body that have picked up mutations, mostly from breathing oxygen, which is toxic, and as a consequence, the DNA is damaged and it's changed, and you have a mutation. We also know that those mutations begin very, very early. We can detect them even in embryos. So now you can ask the question, well, why don't we get cancer more often than we do? And the answer is because we have encoded in our genome hundreds of genes that qualify for being termed tumor suppressor genes. And they converge on a few basic mechanisms that help keep cancer at a low level, at least for half of our lifespan. And those two tumor suppressor mechanisms are a process called apoptosis or cell death. Very clear, a dead cell can't form a tumor. So a cell senses this mutation and dies. The other process is a process called cellular senescence. Cells stop proliferating forever. Also, really simple. A cell that can't divide can't form a tumor. So we think this process, the senescence process, is more important. And the reason is we know that senescent cells accumulate exponentially with the same trajectory of those diseases with age. So everyone here, if you're young, you have few senescent cells. If you're middle-aged, you have more. And if you're old, you have a lot more. The other reason why we think senescence is more important is we see it at virtually every age-related disease we've examined. So that's everything from macular degeneration, which causes blindness, to COPD, which causes lung problems in the elderly, heart disease, and we even see it in precancerous lesions. So these cells are a smoking gun, right? They're present at the right time and the right place to be driving multiple age-related diseases. The question is, how do they do it? And they do it because they're secreting molecules outside of themselves. So here's a cell that doesn't divide. It's not doing anything, but it's secreting stuff. And that stuff that it secretes causes a process called inflammation. And it's inflammation that is the key to understanding every one of those diseases I put on the first slide. So all of those diseases are either caused by or greatly exacerbated by a process called inflammation. And what inflammation does is it destroys tissues, it makes tissues not function well, it prevents stem cells from behaving properly, and it even promotes cancer. So this is the glue. So here's the model. You have a young tissue, and with age, we accumulate senescent cells. We've proven that in the laboratory. These cells produce molecules that they secrete outside of themselves, and it causes neighboring cells to fail to function. And of course, what I told you is that with age, we're also accumulating these mutant cells, and we believe that these secretions can then even drive the progression of late-life cancers. So I'm going to cheer you up. All right. So what do we do about this? So what most scientists are doing now are thinking about two strategies. So one is stop the secretion. So if we can prevent these cells from secreting stuff, maybe we can suppress this driver of aging. And the other is to simply find a way to get these cells, instead of sticking around, get them to die. And both progress is being made on both of them. We actually very much favor this last approach, and I'll show you why. Here are two mice. Now, these are not normal mice. They're mutant. They have a premature aging disease. So we're doing the experiment now in naturally aged mice, but these are prematurely aging mice. They're littermates. And both of them have the mutation that caused them to age prematurely. So mice normally live 2.5 to 3 years. These mice live only 8 or 9 months. They're about 8.5 months now. So these mice accumulate senescent cells at a very high rate, much faster than normal mice. And we can do a genetic trick. So this is only done in transgenic, genetically engineered mice. That's why we can't do it in people yet. But we can get those senescent cells to die. So here's a prematurely aged mouse. It's going to die within half a year or so, or half a year or so, half a month or so. It has cataracts, it has weak muscles, poor bones, and almost no subcutaneous fat. Here's the same mouse. All the senescent cells are gone. No cataracts, no muscle mass loss, no weak bones, lots of subcutaneous fat. Now, these mice died at the same time. So no increase in lifespan, but obviously an increase in health span. This mouse died much healthier, much happier, we think, as much as we can tell that about mice. So the promise of aging research really, and what is our main challenge, is to translate these findings in mice to people. And what we want to do is find ways to eliminate senescent cells in people, provide new stem cells, reduce this inflammation, and maybe even engineer new organs and tissues. All of these on the horizon. What we really want, because we're not sure we can extend lifespan, is to slow down aging. And I will leave you with a quote from Thurgood Marshall. How many of you know who Thurgood Marshall is? Yeah, he's first Black Supreme Court Justice in an era which was slightly less enlightened than our current era. So these are lifetime appointments, right? So someone had the nerve to ask him, how long do you plan to live? Okay, here's my punchline. His reply was beautiful. He said, I plan to live until 110, and then to die from a bullet wound from a jealous husband.
Thank you, Judy. Our next presenter will be Kai Vetter, who will be discussing radiation, something we all need to know more about. All right, sir. Please welcome Kai.
Thank you, Jeff. Good evening, everyone. Kai Vetter, I'm a physicist. As a matter of fact, I'm a nuclear physicist. So we deal with radiation all the time, me, my students, and my scientists. So we're exposed to and volunteer to work with radiation. So we know quite a bit about radiation. So that's why the first question is not really serious. I mean, it's more addressing and maybe some questions you might have about how can we see radiation, because we believe we can see radiation very well with our sensors and our detectors we develop and improve all the time. What is so really the scientific question we also not able to answer fully is how harmful is radiation, particularly at low dose when you have a small amount of radiation over time. What is the effect? So this is really the scientific question we need to answer, we're trying to answer. Now, when we talk about radiation, of course, we cannot ignore what happened more than 4 years ago now in Fukushima in Japan. As a result of the northeastern earthquake, enormous earthquake resulting in tsunami, and then taking down 3 of the 4 reactors at the Daiichi nuclear power plant in Fukushima, leading to explosion, core meltdown, and to significant releases of radioactive material into the atmosphere, into the local environment, into the atmosphere. Enormous impact, socially, politically, economically, locally predominantly, but also globally. I mean, politically, as we all know, has enormous impact due to Fukushima. The question is, what really happened in terms of the health impact due to the radiation? And the answer is, well, I will get to that in a second at the end. But before I do that, a few words to explain what radiation we are talking about here. Because we are talking about nuclear radiation, that means radiation which is originated from the atomic nucleus. And because the atomic nucleus is a strongly bound entity, also the radiation emitted is high energy. That means if you now plot the electromagnetic spectrum, capturing the different types of waves we are exposed to naturally or electively, we go from radio waves, long wavelengths, low energy, to visual light, of course, which we can sense very well. Of course, that's what our eyes are for. Also infrared, we can sense, so we can actually feel it by the heat. And then we go up in energy to the X-rays, and then ultimately to gamma rays. And one of the challenges there, of course, is for us that we are not able to feel it. So we don't really understand what is our environment and what will be the impact of the radiation, the nuclear radiation around us. Specifically, we're talking about ionizing radiation. So radiation that is able to ionize atoms or molecules. That means it can remove or knock out electrons out of the orbitals of our atoms or molecules. So the interaction is quite different than the interaction due to this radiation, which we have to be aware of. Therefore, also in terms of the effects, might be a little different than the effects here. On the other hand, in terms of detection, that might be very helpful to actually help to use the ionizing, the characteristic of the ionizing radiation to detect. Now, one word about radioactivity and radiation, just for clarification. So radioactivity fundamentally just means the transformation of a nucleus from an unstable configuration to a more stable configuration. So unstable to stable, and it can, in to go to the more stable configuration, it emits radiation. And Ernest Rutherford already in 1908, so more than 100 years ago, recognized that there are three different types of radiation: alpha decay or alpha radiation, beta decay and beta radiation, and gamma radiation. And they're all a little different. And they do interact differently, therefore can be also stopped and attenuated differently. Alpha particle, sheet of paper; beta particle, you'd need just your hand or a piece of aluminum. Gamma rays are very penetrating. So if you want to probe and detect some nuclear material in your environment, gamma radiation might be useful because it allows you to detect it in some distance, for example. And then we have neutrons, which I'll not talk about. So that's about radioactivity. Again, the fact that nuclei are not stable, they emit radiation, and we can characterize a different type of radiation. Now, what about the world we're living in? Okay, so what is normal in the world in terms of radiation, specifically nuclear radiation? Alpha, beta, or gamma radiation. So what is shown here is a pie chart reflecting the different sources of radiation we are exposed to in the United States on average, on a daily basis, pretty much. What is interesting that half of the radiation we're exposed to is due to natural sources, particularly what we call primordial sources. That means uranium, thorium, potassium, which have been created maybe billions of years ago in supernova reaction, supernova events, creating the heaviest elements. And their half-life, that means they live for a long time until they decay and emit some radiation. So they have been with us for billions of years, and they will be with us for billions of years. So this is about half of the radiation we expose. Now, the other half is medical. And that really has changed over the last 10 years. Medical procedures, specifically if you look at computed tomography. So enhanced quite a bit more use over the last 10 years, 15 years, now 24%, but overall medical is about half of the total exposure. So not that we really care too much about units and numbers, but it's whatever number, 6.3 millisievert, so that's per year. That's the dose rate for a US citizen in average. Now, if you go to other places in the world, particularly in India and Iran, Ramza, for example, Iran, it goes up to 250 millisievert. So factor 50 or so, a factor 40 higher than we are exposed to on average. So factor 40 higher. That's interesting. Also, some more elective exposures, and I always mention with my students, I mean, what is interesting, that we all are radioactive. We have also potassium, we have carbon-14 in us. That means if we are close to another person, we will be exposed from the radioactivity from the other person. And as a matter of fact, you can measure it, how much it is, which is whatever 0.01 in whatever strange units. But you can measure it, and what is most important, it's not zero. Okay? In our universe, zero radiation is not possible. Okay? There's everywhere, at least in our universe. Has been in the past and will be in the future. Also, what is really important, radiation and ultimately biological effects do not care where it's coming from, whether it's from the natural source or a non-natural source. Okay? It does not matter. Gamma ray is a gamma ray, beta ray is a beta ray. For example, bismuth-214, which is natural occurring, is very similar to the man-made cesium-134, cesium-137, which is the signature for Fukushima. So at the end, the effect will be very similar. We don't care about the origin. So that's really two important messages we have to recognize. We have to, nothing else, that's the takeaway message. Okay. Now, how can we see and measure radiation? Okay, that's one, of course, the first question. Now, if you look at visual light, and we have our eyes, which are extremely sensitive detectors and sensors. Right? We can measure the intensity, how many photons, so like the brightness. We can measure even the energy, that means what color. And we can, of course, image it. We can localize the source of whatever, of the object. Extremely powerful. Now, for our gamma radiation detector, we can do very similar things. We can count the number of gamma rays. We can measure the energy of the gamma ray, which is a very important and powerful tool, because that allows us to distinguish the different types of radiation and their sources, different radioisotopes. For example, whether it's cesium-134, cesium-137, or bismuth. So this allows us to distinguish about the origin or the fingerprint of the sources. And we can also image. Our radiation detectors can be even built more sensitive than our human eye. Because a human eye at least takes about 30 photons in 30 milliseconds to actually see it. Like for our detectors, we only need one particle. They can count each particle individually. So sensitivity is enormous, which is really a big advantage of our detectors. So we can see extremely small amounts of radiation, like single rays or single particles we can see. So also they can be detected before they are harmful at all. So that's a very also important message. So just that I'm not just sitting here in California, so I'm spending a lot of time in Fukushima, trying to help to do better measurements and also help in some of the issues related to the communication about trust. So I've been 4 times to Fukushima last year. So that was the 3rd anniversary. I was in Fukushima City, and I had my radiation dosimeter. Again, that's my radiation detector, which I have always with me, and checking on the flight to see really how the radiation goes up and down on the airplane. And this is in Fukushima City 1 year ago. It's 0.2 microsievert per hour. Whatever that means, if you now add that over the year, it's 2. So it's really the average of the United States. So if you go closer to the site, we go up to 80 millisievert per year. Okay, so it is much higher, but it's still not really that much higher than some areas in the world, right next to the Daiichi nuclear power plant. So this is what we can do with the simple dosimeter, a Geiger counter. Oh, that's just a counter. Now, if we really want to understand where the radiation is coming from, we have to be able to resolve all these different peaks and lines here, because that's what we measure and what we have measured in Berkeley. 1 week after the releases, we are able to measure that with quite sophisticated instruments. Again, you can see the colored ones, that's really what we can associate with the radiation from Fukushima, and the blue ones, the plaque names here is just natural radiation. So having this device will not tell you where the radiation is coming from. You need this type of a device, which tells you really the individual energy, which can you associate with Fukushima or not. So this is not good enough. Okay, going briefly back to Fukushima. And so what have we done in Berkeley? So when it was clear there was some major releases of radiation, we set up our own measurements to look at air, water, rainwater, and many food samples. And we continue to do that. In our redwatch.berkeley.edu as a website, you can find a lot of information about that. So we set out to do the measurements, but also then to make these measurements available to the public right away, and engage the public in a dialogue to try not only to show the results, but also put the results into a context so that the public could understand that. So we were able to see the increase of, for example, iodine-131 in water, and then the decline. The same with cesium in local milk, up, and then the decline. What is important on all these plots is kind of the reference. So this is 0.5, whatever units here is the maximum number we've ever measured in cesium-137 in milk. Now, if you look at milk, we have potassium-40 naturally. It's 50. So 50, so it's up there relative to what we have been able to measure. Yes, we're able to see it, but it's orders of magnitude less than we're normally exposed to. If you just drink milk, and obviously you want to drink milk. We have done that in the past, and we'll certainly should do that in the future and not stop it because of the potassium-40. Okay, so now the question though, is it harmful? Well, this is really a scientific question. And the answer is, well, it depends, obviously. We're scientists. Always the answer is it depends. That's what my wife is always complaining about when I answer. At high dose, we know, yes. It's like with anything. At high dose, particular high dose rate, everything is pretty much harmful. It can potentially. But what happened at low dose? So we are not sure, but we know that the risk to get cancer, the long-term effect, is really small. But we don't really know exactly what's going on at low radiation dose, because the problem is that we have insufficient data. Now, the reason it's very difficult to study a low-dose effect is, of course, the normal cancer rate, the cancer incident rate for humans. Right? It's not 0. It's actually 30 to 40% in the United States. And we are not able to distinguish the cause for cancer, whether it's radiation-induced or has some other reasons. So that prevents us to do these studies in more quantitatively. Now, what the regulators do, and that's really the confusing part, assumes the most conservative approach, which we call the linear no threshold model. That means from the high dose effect, which is linear, they just extrapolate down to 0. And do not consider any effect in terms of time or duration, number of type of the subject of pose, or do not assume any repair or response or adaptation or stimulation process in organism, which we know nature normally does. But we are not able to determine that. Therefore, we use the most conservative approach, which is a big problem. And for us, this is really something we have to improve to better understand low-dose effects. Now, going back to 4 years later, 85,000 people are still evacuated in Fukushima. And certainly, they want to go back. But there are a lot of issues related to transport over the next 20 years out of the environment into the evacuated areas. Now, what is really the health effect? And the international agencies and commissions agree on that. The main effect is not radiation. It's psychologically, the stress, uncertainty due to the lack of information knowledge, the fear of radiation, change in lifestyle. People have to move. They were not allowed to go outside and exercise. So the change in lifestyle was tremendous, and therefore the impact was tremendous. Very likely that no death can be attributed to the radiation due to Fukushima. Okay. Very important message. Really, no death can be attributed to the radiation from Fukushima. However, there are more than 1,500 deaths associated with the evacuation and the change in lifestyle, which is significant. Of course, now the question is, what would have happened if these people, hundreds of thousands of people, would not have been evacuated? Let's leave them there. Get them exposure up to 70 millisievert per year. Again, other areas actually are able, living in that environment. And in the the answer is, well, we don't know. Right? We don't really have the scientific data yet to determine what would have happened to these people when they would have stayed there. They certainly would have been more happy in terms of their environment. They had stayed in the environment, and they were exercising and not change their lifestyle so much. So in order to address the outstanding issues, so what we have formed now is an Institute for Resilient Communities that captures a lot of the research necessary, from the assessment to the prediction to health impact and remediation, but coupling that into the context of education, outreach, and the public communities. And we're doing that by working with communities in Japan. So the mayor of Koriyama City, the largest city of Fukushima, was visiting Berkeley 2 weeks ago, and he spent the whole week with us to discuss how we can help them in terms of doing the work, the technology development, the demonstration science, but also to establish this institute there as a trusted resource. So that's what we're trying to do, to establish an entity, this institute over in Japan as well, to help them to ultimately go back to their homes and live safe in the future. And that's pretty much it. Thank you.
So we actually have 6 questions since Kai used an extra 10 minutes, but that's okay. That's okay. Yeah, that's okay. So we now move from radiation to insects, and Javier Navarro, please give him a warm welcome.
Okay, so there it is. Again, my name is Javier Ceja-Navarro, and I am a chemical engineer working with insects at the Berkeley Lab. One of the main questions driving my research at the Berkeley Lab is, do insects contain the secrets for food and energy production, for a sustainable production of food and energy? To be honest, I didn't really care about insects and about their beauty before. And then everything changed when I came to work at the Berkeley Lab with this amazing system. I caught the bug, okay? So after this presentation, I hope that you will get a little bit of an insight of our research, but also that you will get to change your view about these amazing creatures. Have you ever considered that insects are everywhere? They feed on all kinds of food. They are in all kinds of environment, from the desert to the tundra, soil, water, plants, even on animals, including humans. Just look at that beautiful loss hanging there in the hair of a human. So what's the underlying reason behind this versatility of insects? What is giving them the ability to adapt and to survive to pretty much all kinds of environments? Let me tell you a little bit about it. Insects represent 56% of all living organisms in our planet. Okay? They are the most diverse group of organisms as well, and there are estimations that we have around 10 quintillions of insects alive at any point in our planet. That's a 10 with 18 zeros. Okay. So usually when we think about insects, we will think about a pest. Okay? And immediately we will get the image of a roach in our mind. Yeah, some of them are destructive. They can attack crops or even spread diseases. But most insects are harmless. Okay? They can provide a lot more to an environment and to humans, and we forget about that. For example, we can get a lot of natural products from insects, including wax, for example, silk, honey. They are also models for scientific research. We can study genetics or even study animal behavior. They are also agents of pest control and pollinators. In fact, without the activity of these pollinators, many plants would disappear from our planet. And with them, also the animals that depend on them. Also, something important about insects is that they are ancient creatures. Okay? They appeared on our planet 300 million years ago, just 100 million years after plants. And we humans, well, we came here 250,000 years ago. That's nothing. So who do you think, or which multicellular organism do you think has developed better strategies to live in this planet, to interact with the planet? Insects had millions of years to create the strategies to interact with plants, with other microbes. And while doing this, they were able to incorporate some of these microorganisms to the digestive system. There, while giving them this really nice environment in their gut, they will get the benefit of all these microbes and their abilities to transform all kinds of substances. So, could we benefit by studying the perfected biology of insects? Okay. Let me tell you about the systems that we're studying at the Berkeley Lab. One of them is soil microcreatures from a soil that has really low content of carbon, nitrogen, and phosphorus is in a form that is not available for plants. Soil microcreatures and other soil animals, they move through the soil, they modify their structure, they allow air to go through the soil, nutrients, and water. But how about the microbes that are associated with these little animals? Could they be also contributing to the cycling of carbon, nitrogen, and making this phosphorus available? My other system is the passalid beetle. This is an amazing creature. Okay? These are subsocial insects. They have no hierarchy. They are all the same. They have really strong family bonds, and they interact by making sounds or chemical signals. Something that is really important about these creatures is that they can degrade wood. They are really good at physically and chemically transforming wood. In less than 1 year, 5 of these little creatures would transform 5 kilograms of wood. So that's a lot of material. And while doing this, they are decomposing the wood, its polymers, including lignin, which is really hard to transform. And during that process, they will get all the energy they need to survive, to live. So could we harness this potential and maybe learn about it and use it for our benefit? My third system is a coffee pest. It's known as the coffee berry borer. This little organism that I'm showing you here evolved together with coffee plants to the point of making coffee seeds its only source of food and shelter. Okay? So then, when coffee got out of Africa, of course, did the coffee berry borer. It was following the plant. It's home. But the amazing thing about this creature is that while living in the coffee seed, it exposes itself to toxic levels of caffeine. And just to give you an idea, the caffeine needed to kill a human is 0.075 milligrams per gram of body mass. That would be like 50 shots of espresso for a guy of 150 pounds, maybe. So this little insect lives in an environment that has 100 times or a little bit more caffeine than they're requiring to kill a human. How is this guy doing this? Could we use this ability maybe to control it better? Okay? The Berkeley Lab is a unique space, a unique place where multidisciplinary research gets combined to study well, all kinds of problems, health, environment. And at the Earth Sciences Division and the Ecology Department, we have a set of experts that apply omics approaches, such as DNA sequencing, RNA sequencing, or even the application of different analytical techniques to study the environment, being soil, water, plants, and now also insects and their associated microbes. For example, here, what we did with some of these insects was to extract their DNA, their proteins, do sequencing, and identify all the different organisms living inside of the insect. But not only this, we're able also to elucidate the steps that occur in the insect for the transformation of different substances. For example, here, for the passalid beetle, this is an image of what its digestive system would be. Okay? Once the wood enters to the beetle, it will go to the midgut. There, the polymers of wood will get transformed into simple molecules that then go to the anterior hindgut where fermentation occurs. All the energy is then produced, and then you have an extra step for degradation for whatever went through. Okay? Another thing that we do at the Berkeley Lab is to analyze the gut environment, the digestive system. How is this environment? How are these bacteria living in this internal environment? We require this if we want to reproduce this outside of the laboratory. For this, we are using different approaches, as I told you, like microelectrodes to measure the oxygen in the animals, or even CT scanning, like what I have in here. And with this, what we want to do is to understand how the gut is arranged inside of the little creatures. How is this little refinery arranged in the animal? Once we understand this, once we understand the environment, we know what these different organisms are doing inside of the insect, we can try to take them out. Here, for example, you can see several microbiology plates with caffeine, cellulose, and lignin. And the dots that you see in there are bacteria and other organisms that are able to survive using only these substances that maybe we could use later for other approaches or for other applications. Okay, so what have we learned so far? From the soil microanimals, we know that they participate in the recycling of nutrients. So could we use this knowledge to generate new soil management strategies? About the coffee insect, we know that this little creature survives in coffee because of its bacteria. So how about we target that bacteria to change the preference of the insect for the coffee seed, and maybe it moves out a little bit to the fruit, so it doesn't really affect coffee production? Could we use this idea of targeting bacteria to control other agricultural pests? About the passalid beetle, here we have an amazing biorefinery. How about we mimic nature, and we learn about the processes, and then we create new approaches to produce energy? Humans, well, we have become really good at reproducing and living longer. And while doing this, we are exploiting our environment without really taking care of doing this with a balance, with sustainability. And this will take us to the point of being unable to provide the resources for future generations. You know, at this point, well, we are able to travel to space, to analyze genomes, to analyze huge amounts of data. How about we start paying attention to our environment, to its creatures? The key for a sustainable existence in this planet may be out there. We just need to learn from nature. Thank you.
Thank you, Javier. What you did not know is that Javier deserves a second round of applause because he is responsible for the drawings in his presentation. So next up is our little math moment. I think there's only one equation in there which is sort of irrelevant, right, Ann? No. Well, no. Okay. I'll let Ann explain it. It's about, do we need math to blow up a star? Please.
So Javier is a very hard act to follow, so I'm going to answer this question, then you don't have to vote for it, and I won't feel bad. So the answer is yes, we need math. And so what I want to do is spend the next 10 minutes convincing you guys. Oh, he didn't start my clock. I get extra time. Okay. So what I want to convince you is that We have microscopes, we have telescopes, we have computers, we have detectors. Yet we still need math. And yes, this is the math that you learned in elementary school and high school and ... And I want to try and convince you that it's important. Okay, math in the context of a supernova. Since Jeff got to talk about the origin of a word, I get to talk about the origin of a word. So supernova is super plus nova. Nova meant new star. We looked up in the sky, there's something bright there we didn't see before. Aha, it's a new star. It's a nova. Well, then there's one that's even brighter. It's a supernova. And you don't actually have to travel that far out in space to see one. In fact, does anybody recognize where this is? Okay, so there was a supernova in Oakland. This is if you are getting on 580 from, I think you're on Lake Park passing Grand, there was a billboard up there. And if you know scientists, we all love to get published. We want to see our names in print. But seeing your picture on a billboard is way cooler. This picture up here is of a supernova. It's before an explosion. And it was made with a code that we wrote at Berkeley Lab. So that's the supernova in Oakland. Unfortunately, they took it down. So this is what one usually thinks of as a supernova. And this is in a galaxy far, far away, in fact, about 60 million light years away. And this picture, it's a classic picture. If you go on Google, you will see that everyone puts up this picture of a supernova. It's SN 1994D, so you can guess what year it was discovered. And the three things you need to know. So one is we don't see them usually by looking up in the sky. This one's actually an image through the Hubble telescope. Two is, and I don't have a pointer, if you look in the middle of the picture, that's a galaxy. A galaxy is many, many, many stars. You look at the bottom left, that is one star. So a supernova has the ability during its lifetime to be as bright as an entire galaxy. So that's a pretty amazing thing. And then the third thing about them is they're only visible for several weeks, kind of like my billboard. Okay? So why do I care? And I have to say that Kai stole a little bit of the thunder because he used the word supernova. And if you remember what he said about them, he said, that's where all the heavy elements came from. So in the Big Bang, in the beginning, we had hydrogen, we had helium. We didn't have all these other elements that we have that Berkeley Lab discovered some of. And so all of these other elements came from supernovae. So if you ever hear the expression, you're made of stardust, it's real. That's what we're all made of. So here is a brief history as to why you should care about supernovae. So if we look back 1915, 100 years ago, Einstein said the universe is stationary. Oh, he used the word universe too. You killed all my punches. So the universe is stationary. The universe is space. It's the stuff that surrounds us. It's not getting bigger. It's not getting smaller. That seems pretty reasonable. We're here. We've been here. We're going to be here. Well, Hubble came along. He had some telescopes. He observed galaxies, and he observed that all galaxies are getting farther and farther away from all other galaxies. And there's only one way for that to happen. It's not just that something's leaving me, but I'm getting closer to something else. Everything's getting farther away. And the only way that happens is if the universe itself is expanding. So we started the Big Bang. We're getting bigger. This sounds great. Okay. Einstein actually admitted he'd been wrong, which I love because everyone thinks Einstein is the god. He was a really, really smart guy. He was wrong. He admitted it. We move on. That is a great way to do science. Okay, so now, looking at more galaxies, one of the things they noticed, you can look at the rotation rate. So galaxies spin. They don't just sit there. And just the same way, if you look at the Earth going around the sun, if you know how far it is from the sun and you know how fast it's going around the sun, you can calculate the mass of the sun. Same thing with the galaxies. Galaxies spin. They rotate around other things. If you look at how fast they are, how far away they are, you can figure out how much mass is in the middle. And the problem is that there's a lot more mass in the middle than we can account for by adding up all the things that we can see. So what that tells us is there's a whole lot of mass out there. There's a whole lot of matter. So mass is matter. Dark means we don't know what it is. Okay, that was the first thing I learned was, what's the secret? What's dark matter? Dark is we don't know. Okay, so now if you like to worry, and I come from a family of worriers, you say, wait a minute, mass is gravitationally attracted. Mass always attracts. If there's a whole lot of mass, maybe it's going to suck the universe back in. We started in the Big Bang. We're headed for the big crunch. So if you want to stay up at night, you can worry about it for about 2 seconds until I tell you, no, you don't need to worry. You can go home and sleep tonight. So 1998, Science Breakthrough of the Year. This is actually Science Magazine. This was on the cover. You can see the expanding bubble that Einstein's blowing out of his pipe. How did they figure this out? They said, well, we've observed a bunch of type 1a supernovae. We're going to make some assumptions about how they behave. And we are going to conclude that the rate of the universe expanding is increasing. So not only is the universe getting bigger, it's getting bigger faster and faster. Okay, so now you can sleep at night. This is good. I haven't said math yet, so what's going on here? So it's this middle part that bothers me. Now, I have to tell you, I'm a computational scientist. I'm not an astrophysicist. So everything that I've told you I had to learn not that long ago. And I said, well, these scientists, they must know what they're talking about. Well, whenever you see the word assumptions, you say, okay, what did they assume? We'll talk about that. So let's talk about how do we know what supernovae do? If we're going to make these assumptions about how they blow up, how do we do that? Okay, so you observe them. Well, you observe them. Here's the Hubble telescope. We talked about that. And what you see is only the light from the supernova that hits the Earth. It's the observables. It's what you can observe. This picture, on the left, you can see a star that is dark, and then it gets bright, and then it gets dark again. Upper corner is just tracing out how bright it is. And then the lower part is telling you that we actually measure in different colors. We don't just measure white light. We measure in different colors. That's what you observe. So how do you study a supernova? You can't get up close and personal. You can't send a probe into a supernova. And this is the 3 pillars of science, or the 3 rings, kind of Olympics looking. Experimental / observational, theoretical, think pencil and paper, computational. So we have computers. This is great. I still haven't said math. So here's the point. We're going to use computation. Okay, so we have computers, end of story. And here's where it comes in. What's a star? Okay, we've got to put a star on a computer, and a star is a fluid. Okay, water is a fluid. Air is a fluid. So we can use the equations of fluid dynamics to model a star. Okay, this is great. Whoops. Fluid dynamics describes 3 things. It tells you how the fluid moves. It tells you, you pour the glass of water. Water goes from the glass onto the floor if I'm pouring. It tells you nuclear burning. This is how elements become other elements. That's how hydrogen and helium became all these other things, how carbon and oxygen became magnesium. And fluid dynamics tells you sound waves. So sound waves are the reason that you can hear me now. If there were no sound waves traveling in the air from me to you, you would not hear me. The equations of fluid dynamics do all 3 of these things. So we have a supercomputer. I think we even have one of these pictures. These are supercomputers that are actually at Berkeley Lab. If you put these all in a supercomputer and you use all the processors you've got and all the best techniques you've got, you can model an explosion for about 2 seconds. And that's just the explosion itself. And the problem is, if you ever study fires, like house fires, forest fires, any kind of fire, the outcome of the fire often depends on where it started, where and how. In the 2 seconds, you can't get there. So we need about 2 hours. And it turns out that those equations I showed you, we can't do it. We can't use those equations and do more than about 2 seconds. So finally, I get to my punchline. We need math. Okay? We want to figure out how the supernova starts, which is going to tell me how it blows up, which is going to tell me whether it really is the same as all the other supernovae. We need math. And it turns out, and this is the part I'm skipping over, if you take out the sound waves, you can solve it a lot faster. And the point is, if I were holding a match up here and somebody asked a question, it doesn't change the burning of the match. We can neglect the sound waves. And so we use our math, ... to say, let's come up with some equations that don't have sound waves. And you guys had probably already figured out that this is what they would look like. So this is what they would look like. And it turns out that computationally, we can solve these a lot faster. We know how to do that. And so now we can do the full 2 hours. And let me just see if I can get the movie to start again. So here we have, this is the nucleosynthesis in an exploding star. This one happens to have ignited at the very center. Not all of them do. And so I'm a computational scientist. I love this. I love that we made progress. I won't say solved, but we made progress on the supernova problem. But what's really fun for me is we can use those same ideas. We can use those same mathematical techniques, those same ideas, and actually those same computational skills. This is cloud formation. This is rising air. It's moist air. This is liquid water. We just made a cloud. We can use the same techniques to model a burner in a laboratory. So we have colleagues in a different building. They actually burn things. We put it on a computer. We match it up. And it's surprising what we learn. And that, to me, is the power of math. Thank you.
I am a believer. I'm a believer. And she did that under 10 minutes. Next round of applause. Okay, our next and last presenter, this is going to be a tough vote, I think. Shashi Buluswar talking to you about sustainable urban food production. This question, please welcome Shashi.
Since the dawn of the agricultural revolution, food has been produced a particular way. And the way it has been produced is that it's in rural areas. And if you think about how forests grow and evolve and die, essentially they operate on their own cycle. They don't need any intervention from us. We've changed that around in our food production systems. What we do is we suck more out of the earth than the earth is capable of giving. We pump fertilizers, pesticides, herbicides, and so on and so forth. And as a result, we've devastated the planet. This question was not relevant a few years back. Nobody cared about sustainable urban food production. And we'll see why it's more relevant today than it was. And then we'll see whether or not it's possible. And sustainable means a number of things. It has to obviously work for the environment. It also has to work financially. What we'll see is that the reason urban food production doesn't work today is that it is too expensive to be financially sustainable. So behold the mighty tomato. Why should we behold the mighty tomato? It is, if you think of what it does for us, it is almost the perfect food. So if you think about how much actual caloric food value we get out of it and how much nutrition we get out of it, it is right there in the top corner. Compared to, for instance, nuts, very nutritious, but you have to eat a lot of nuts to fill yourself. And then, of course, you have chicken McNuggets, other wonderful things that our food production systems have given us, right? Which fill us up, but are not very nutrient-rich. Now, what does that have to do with urban systems? In a city like Oakland, one of the things we see is that the poor don't have access to good food. Right? And the way that manifests itself in what people eat is this. So if this is essentially what low-income people eat versus higher-income people, and what you see is that the only thing that lower-income people eat more of is sugars. And this is not just a scientific issue. This is an issue of economics. There's a reason why there is no Whole Foods in West Oakland. Right? Whole lot of considerations that food companies and grocery stores have to go through. And as a result of that, if you are a working parent working more than one job, you come back, you're really tired. You can't actually go the extra mile to get good vegetables and cook. And as a result of that, you see the health consequences. So not surprisingly, lower-income Americans are more obese and suffer all sorts of diseases that higher-income Americans don't. Now, some of these pictures are not from Oakland, but might as well be. The food desert issue is a massive one, which is that in virtually every inner city in America, you cannot get good food. You get instead this kind of stuff, right? So this is an FDA map of Oakland, and there are a number of categorizations of food deserts, right? The yellow and the orange and the red. And guess where the McDonald's are, right? It is not surprising that the places where you get Whole Foods is not the places where you get McDonald's. So that's the one problem about access to nutrition. The other problem is about the environment, which is we produce ridiculous amounts of fertilizer, which fertilizer unto itself is a very carbon-intensive thing to do. It's also devastating waterways. What we do is we pump all these runoffs into our rivers and oceans, and we're killing off sea life. Then another problem that nobody in Oakland needs to be reminded of, nobody in California needs to be reminded of, which is water. We'll see how much water it actually takes in just a bit. In addition to that, you have pesticides, herbicides, all sorts of things that we do to suck, as I said earlier, more out of the earth than the earth is capable of giving. And then, after we do that, we ship stuff far, far, far away. Not as far as the supernovas we're talking about, but certainly it does not make sense to grow stuff in California, and ship them to Michigan, as good as that is for California's economy. Now, you add all this together, what does that mean? So if you take the tomatoes, the mighty tomato again, consumed by the city of Oakland, here's how much its footprint is. It is 150 million gallons of water. So all the tomatoes in the city of Oakland in 1 year. It is, 9,000 gallons of fuel to transport the stuff, and just a ridiculous amount of pesticide and herbicide. So let's come back to our question. Is it possible to grow food sustainably, locally, and cost-effectively? Well, when we think of local food, this is what comes to mind: community gardens. And the problem with community gardens is this. They are well-intentioned, but they often depend on charity. They often depend on subsidies. And so what you see is for every dollar you spend on things like tomatoes in typical conventional rural farms, if that were grown organically in rural areas, it would cost $1.50 or so. If it were grown in community gardens, a lot more, conventional or organic, right? And so this is why organic farming is not financially as sustainable, and community farming is not financially sustainable. And so the question is, how do we then make this more cost-effective? The other part of this equation is that we're solving for the transport problem, but it's not clear that local food production is actually environmentally that much more sustainable, because who knows how much water is going into it? Sure, usually community gardens tend to not use fertilizers and pesticides and so on, but there's no law against it. And honestly, if these chemicals were used in a city, it'll probably do a lot more damage than if it were used in a rural setting. So now, if you think about the various ways in which food production systems have evolved, historically it's been here. It is just using soil. So the mediums being soil, some sort of a substrate, and then you get more and more sophisticated, and we'll talk about some of the more sophisticated systems in a second. And then you have just a regular old outdoor farm. You have greenhouses, and then you have what I'd like to call a hyper-controlled environment. So greenhouses have been around, and they're getting increasingly sophisticated. And I believe that the really interesting stuff is up there, where you can super control the environment, so that instead of the old agricultural systems where we had to pump stuff into the environment to fight off the harshness of that environment, all of a sudden we're making the environment much more conducive to growing. So in other words, imagine food production systems where if it's in a shipping container, where things are pretty well controlled. And we'll talk about what those controlling mechanisms are. So rather than having a farmer who really has to understand the soil and insects and the rain patterns and so on and so forth, all this stuff sort of gets automated and controlled. Right? A little bit like those cappuccino machines where my cappuccino making skills are no longer relevant, which is very sad, because you can press a button and get stuff that's okay. So this is what we're trying to do, is precision agriculture. So three systems. The first is hydroponics. Essentially, the idea is that instead of the roots being buried in the soil, it's in water, and that water keeps getting circulated. The water has nutrients, and because of that, because you're reusing that water, you reduce the amount of waste. Right? Then you have aquaponics, which is much, much more interesting, which is you actually have now in that water fish. So the fish poop, which my daughters get a kick every time I say that, actually generates nutrients. Obviously, you can add additional nutrients as well, but now you're starting to get sort of a closed-loop system where you don't have to add a whole lot of new stuff. And then you have a much more interesting, much more sophisticated mechanism called aeroponics, where you're actually spraying stuff onto the roots, right? Where the roots, as you see, I don't know if you can see in that picture, but the roots are no longer, they're not buried in anything. They're suspended in the air, and you're spraying water, you're spraying nutrients into the roots. Okay? Now, if these systems are used at scale, and already a number of people are starting to do this, there's a tremendous savings in terms of water. For some of these things, you can get by with 5% of the water, a lot less of the fertilizer. And because these are now in controlled environments, indoors, in greenhouses and such, you're not exposed to things like pests as much. So you don't need as much pesticides. And a lot of the systems that we're looking at now, they use almost zero pesticides. And then the challenge now is even these systems, and there are a number of innovators that are growing vegetables this way. Now, many of these folks are scientists who've taken off their day jobs and starting to become farmers. Even though they're doing really interesting stuff, it's still not financially feasible. For every dollar, again, it's costing them $1.25, $1.50. And that's a fundamental challenge they're trying to overcome. And the reason is that if you look at this greenhouse, a lot has to go into the actual structure. The glass is expensive. It gets dirty. Lighting. There's energy costs involved in lighting and heating. And then hyper control. So for instance, if you go into one of these systems, one of these greenhouses, you have to wear this suit. Otherwise, you're actually bringing in pathogens into the system. And this gets expensive. How do you make this less expensive? Then the other problem is that, interestingly, a lot of these folks are not growing tomatoes. They're growing lettuce and greens because these are single cycle. Right? If it's a single cycle, you don't have to worry about pests coming in the second and third cycles, whereas tomatoes, on the same plant, you have multiple cycles of fruits. Right? And so they haven't cracked the code on how to grow things like tomatoes yet. And then the other thing they're struggling with is that because the science is not yet known, it's all experimental right now. Right? They're tinkering with this. These are really interesting tinkerers, but nonetheless, they are tinkerers, and they don't know the answer, and sometimes the losses can be devastating. And so that lack of science is actually really hindering the development of these systems. Now, what kinds of things can we do? Turns out that plants don't need as much sunlight as they get. Some studies show that you can actually do strobes, maybe even 3 or 4 seconds out of every minute, and that could be enough for plants. And what we don't know, again, is there's some early science indicating this, which is you can actually control the wavelength and actually get more out of the plants by exposing them to some wavelengths than others. And again, a lot of research has to be done here. Then you have this notion of a nutrient recipe. Right now, we use a baseball bat to grow plants. We just throw everything at it, whereas we don't need to give that much or even the same generic mixture of fertilizers. Right? So understanding what crops need exactly what and giving them exactly the precise amount so that not only are we wasting less, not only are we creating less waste, but the plants are actually better off. Because as you know, you can over-fertilize the plant. And by the way, this picture isn't clear, but there's actually the possibility that we can use local waste to do that. And the challenge with local waste is that if you go into a compost bed, you have no idea how to make that precise. Right? So again, a lot of research has to go into whether or not you can take existing waste and make that more precise. And then, as I mentioned earlier, there's this level of hyper control where instead of having to expose growth and your produce to the elements, now you can sort of I'm not exaggerating. I'm willing to bet that 10 years from now, in iPhone version 46, you can actually sort of control what kind of light, what kind of humidity, what kind of temperature your plants get every minute, and you get really interesting sensors in the ecosystem so that there's this feedback loop. Right? So all of this is quite possible. And then the other thing that can be done with things like greenhouses is instead of the traditional glass, can we actually have interesting structures so that now the glass is it is less resistant to things like dirt. Dirt, as you know, occludes light. Right? So a number of really fascinating things need to be done, but currently the food production systems don't lend themselves to this kind of research. So we are actively looking into some of these questions. So let's go back to the original question. Is sustainable urban food production possible? What do you say? Well, I'm a scientist, so I'll only lie to you when I can prove it with data. So I've only talked about the mighty tomato and the mighty lettuce. What I haven't talked about is the real stuff that really consumes a lot of the resources. Right? So I think we can grow vegetables, and that can be very sustainable. But at the end of the day, the grains are what consume most of the resources, whether it's water, whether it's fertilizer, pesticide, and so on and so forth. And I must say, I'm scratching my head about a solution for the grains. Vegetables, yes. Grains, maybe. We'll see. Thank you.
In fact, we might take a question or two while the votes are being tabulated, so we don't want to keep you folks any later than necessary. So let us start right here.
So why is the radiation in India and Iran so high?
You're on.
Okay, very, very good question. It really depends on the environment and the geology of the environment. So they're in these specific areas in India and Iran, they're close to some uranium mines, for example. So they have a lot of uranium or thorium in their environment, causing a lot substantially increased background levels of radiation.
I actually think that Indians tend to have more skin-to-skin contact.
Bravo.
I don't think that has been really proven scientifically yet.
We have the population to prove it.
The density, maybe.
Would it be because of man-made mines, or would it be natural? So is it a man-made higher level of radiation because of the uranium, or is it just because it's like on the surface?
Well, it's nature-made anyway, but it's not man-made. They're the natural mines and natural occurrences of different geology. So it's really not any impact of humans on the creation of the additional radiation. But again, and that's a very important point. It doesn't really matter in terms of the impact of radiation, whether it's man-made or really natural. I mean, the radiation is all really the same, whether it's beta, gamma, or alpha radiation. So it wouldn't really matter. But in this case, certainly it's natural.
Okay, thanks for your question. We'll take one right here. Hey, could you please tell me who it is?
It's for two people.
Okay.
This gentleman referred to sustainable growth, and you alluded to sterile environment was better for growth. I just want the opinion of the insect person, how insect could also promote
very
growth.
good. Excellent.
For clarity, it's not necessarily sterile, but it's controlled. Right? So there obviously you want the healthy ecosystem and keep out the things that are destructive. And he's absolutely right, whatever he says.
Well, in nature in general, we have food networks. And when we're going to work about the design of any new strategy for food production, we need to take into consideration that what are going to be the different trophic levels that must be included in the system, this being even a worm or insects or even the bacteria that you could add to the system together with the plant. Yeah.
Thank you for the question. Take the next one here.
My question is for Mr. Ceja-Navarro. Sorry if I'm saying it wrong. First of all, respect on your socks. I don't know if you guys can see this, but his commitment to research, he's got insects on his socks. So you were talking about your research relating to energy production, and I'm wondering if that is cellulosic biofuels, second generation, going from beyond corn to sort of waste materials.
You know, at the very beginning when we started with the passerelle beetle, the wood-feeding beetle, we were considering only the possibility of the production of, for example, hydrogen and methane, which are produced in the beetle like in a natural way as it happens in termites. So what we were trying to do there was to harness specifically these kind of processes. But then over the time we discovered there that the insect was also, together with the microbes, developed this ability to degrade lignin. So you know that this is a big problem for the production of energy from crops. So then we could also target that part in there. And then because of how the system is arranged in the beetle, you could even use the whole train of processes to go directly for hardwoods, which are really abundant. I don't know if that's...
Yes, thank
okay.
you.
Yeah.
Thanks for the question. Next one over here.
My question is for Mr. Buluswar. Is the marijuana industry in Colorado using your techniques, or are they is that what they're doing in those huge warehouses?
The marijuana industry has been on the forefront of this actually in many ways. The difference is that from a purely economic competitiveness point of view, they're not competing against a heavily subsidized system yet. And because it's a niche market, because they've actually been able to charge a lot, they haven't had to worry about the economics as much, right? But they have, obviously, because of the legal constraints and so on, they've had to be quite innovative. But that's secondhand knowledge.
I think her question was the same as mine. Was it about the marijuana industry?
Sorry?
I think her
Yes,
question was the
that's
same
correct.
as mine.
That's correct.
So since it's my understanding, again, secondhand, that these industries are for the time being legal, have you thought of partnering with them to fund your research?
What a novel idea. We'll take that to the director.
As an employee of a federal government lab, I'd have to think carefully about that. No, but the truth is that a lot of the folks doing this actually were very innovative, right? It'd be foolish of us not to pick up on that knowledge. So the question is, again, just as some of these systems work for single-cycle greens versus tomatoes, part of precision agriculture is really understanding the difference between various types of crops. And marijuana is a weed, right? And tends to be more hardy. The question is, will those techniques then translate to non-weeds like tomatoes and so on? But I'm assuming it's a weed, right?
Yes, it is. So I'm going to interrupt because I have a question for Judy. I used to write about age-related diseases a lot. I'm curious, there's been a lot of research in this area for quite a few years now, but I'm wondering how soon before there's going to be something tangible and of immediate benefit to folks in their daily lives.
For health span?
Yes,
or
health
lifespan?
span. Health span.
Okay. So for health span, we're there.
Yeah, okay.
So for example, statins, blood pressure lowering medicines. So the amazing thing is we now know that these medicines, which are designed for a very, very specific problem, are having broad effects. And so what they're not complete because there's still lots of other diseases that they don't hit. But for example, women who have been on statins have a lower rate of breast cancer, totally unexpected. And so there probably are prototype medicines already out there. We need better ones and we need broader acting ones, but we're on that path.
And how about lifespan?
Yeah, so lifespan, the only intervention that has been reported to consistently extend lifespan in multiple creatures from E. coli all the way up to monkeys is a regimen called caloric restriction, which I don't recommend. It's lowering calories with no malnutrition. So you have to take lots of supplements to lower your calories by about 40%. It's not clear it will work in humans. It's controversial that it even works in monkeys. And what most people feel is that it doesn't really extend lifespan, it just feels that way.
Very true. We'll take a question on this side. Yes, sir.
Okay. Sorry, gang, I noticed this mic was getting kind of lonely. But my question is for Shashi and Javier. I apologize for calling you by your first names. I don't want to butcher your last names. How do you see you two working together? Because I noticed sustainability is the focus of both of your work. And I see a lot of corollary. I'm not sure if you guys see it like that, but I'm just wondering, do you guys see yourself having a future together? Not necessarily, together, but your work, and moving toward, the same goal together, because it really seems like you're working toward the same thing pretty much, the end result.
Great question. And one reason we've taken on this issue of sustainable urban food production is that it requires that kind of multidisciplinary approach that few labs in the world can. Right? And so we're in the very early stages of this, and I'd be shocked if we didn't work with Javier and his colleagues on this.
Yeah, one of the objectives of our research is also the discovery, for example, of new bacteria that you can maybe then add to a system, and then they will help with increasing nitrogen content or transforming carbon. So for sure, there are points where we can collaborate. And these are already things that we are considering in the laboratory that are actually already happening. So, yeah.
Okay, can we take a pause? We have the results of the voting. Okay, let's turn around and let's see. Let's see what we have. Okay, it's very exciting.
Whoa.
Well, is that a factor of the age of the audience? I wonder. It's a demographic issue here. Okay, Judy, congratulations. As I said, there's no prize, but congratulations anyway. All right, next question, sir.
This question is for Dr. Almgren. I had just read an article maybe a month ago in some scientific publication that they can use sound waves to put out flame. So there's some guys that created, and so I was just curious, given that you were saying that that doesn't impact the explosion or anything like that, that you can pull it out. So I was curious how you can subtract the impact of sound in order to look at the rest of it, given that impact.
Right. So what I did is I glossed over a lot of detail in the context. Yes. So in the context of supernova explosions, it's actually not that sound waves are irrelevant. It's what happens. They move so much faster than the fluid. They equilibrate on a time scale that we can resolve with a different set of equations. But you're absolutely right. There are cases in which acoustics are incredibly important. And in those cases, we would never use the alternate equations. So it's a matter of identifying, do the sound waves matter in this particular example?
Cool.
Yeah.
Thanks a lot.
But
Thank you.
Very
question.
good point.
Next question over here.
Yeah, thanks. I had questions related to the mice and the aging experiments. Just first, did the treatment reverse the aging process or just halt it? And secondly, if it wasn't aging that killed the mice at the same time, what was it that killed them?
Yeah. So remember, these mice are premature aging mice, so they have a defect. And that defect causes a heart disease. So whether or not they had senescent cells killed off, they still died of the heart disease at around the same time. So that was no increase in lifespan. What was the second?
Well, just you showed the two mice, and one was healthy and one was older and sicker.
Did it reverse?
Did it reverse or just halt?
Yeah. So in that particular experiment, it was starting with the equivalent of what would be a 20-year-old human. So youngish mice were treated then every few weeks with a drug that would cause them to lose senescent cells. The reversal, those experiments are ongoing right now in several laboratories, and I can tell you they're promising for some tissues, but not all tissues. So it will probably work for some organs, but not for others.
Thank you.
Yes, next question here.
Yes, hi, Dr. Campisi. I had a quick follow-on question as well. I really enjoyed your talk on aging. And if I understood the triggers of events, it starts off with senescent cells, which can cause or lead to inflammation, which can lead to all kinds of maladies. I hear a lot about reducing inflammation as a good strategy, and I'd like to know, is there evidence that focusing on that part of the process has the highest return on quality of life?
So it does. So as you know, the American Heart Association now recommends baby aspirin every day. And I think there's really good epidemiological evidence coming out that shows that, you know, this is an anti-inflammatory, and it does reduce the incidence of heart failure and heart attacks. What I will say, though, about the over-the-counter anti-inflammatories, so aspirin, ibuprofen, is they're incomplete. So we've done experiments with these senescent cells. They suppress some of the things they secrete, but not all. So we do need better, broader acting drugs, and many of us are working on that.
Thank you.
Thanks for your question. We're going to take this question here, and then those folks that are in the line, those are going to be our last ones for the evening. So please.
Question for Kai. One of the solutions for the problem of carbon fossil fuels is some people are proposing more radioactive nuclear reactors, and some of those are putting forward thorium as the panacea for the problems of uranium, plutonium. You have some comments about thorium versus those more traditional fuels?
Yeah, it's certainly been explored as a possibility. I mean, certainly India is being used as their own resource. And research is certainly considering thorium for some advantages, but also for some additional challenges. Also, some of the challenges you might have with uranium might also have with thorium as well. So it's part of more, except in India, there's still more research than in this area of thorium reactors.
Thank you for your question. Yes, sir.
Yeah, my question is also about the sustainable growing. And are you guys looked at, could you expand more on why grains are harder? And also, are you guys looking at options like teff that are a little lower water and maybe more space efficient?
Yeah, I mean, grains just purely because of the volume. Right? Just how much we consume much more weight and volume of grains than vegetables. The thing with teff is this seemingly miracle grain that's consumed in Ethiopia. The one of the hardest things to do in human behavior is change what people eat. Right? So rather than try to advocate for behavior change that way, and it's not mutually exclusive of that, right? We try and see if the things people are eating right now can be made more sustainable and cost effective.
Thank you. Next.
Question about senescent cells. I have quite a few friends who have the autoimmune diseases, Crohn's and neuropathies and rheumatoid arthritis. Do senescent cells play a part? Has that been studied?
So it's interesting. So rheumatoid arthritis is primarily an autoimmune disease. It's not age-related. I mean, it does happen in very young kids. It's a little bit rare, but it's primarily a problem of the immune system. So far as we know, senescent cells do not play a role in that disease. Osteoarthritis, on the other hand, is purely age-related, and there's lots of evidence that osteoarthritic joints are loaded with senescent cells that form the cartilage. So it depends on the type of arthritis. Things like Crohn's disease or irritable bowel disease, those are also driven by inflammation, probably not due to senescent cells though, probably due to leakiness in the gut, and so the bacterial products in the gut then elicit an immune response.
Thanks for the question. Let's take yours first.
Okay, I'd like to start with math and blowing up the stars, the supernovas. You showed us a type 1A supernova. What type of supernovas are there? From there, I'll segue into radiations. What are the types of supernovas that are observed that we know about?
So the main types are type 1 and type 2. So they keep it simple. Within the 1, there's 1A. So there are subcategories of those. Type 1 and type 2 are fundamentally different. So, right. So type 1 is a thermonuclear explosion, and that's so the type 1A is a specific one. It doesn't have a silicon line. They get into subtleties of the explosion, but type 1s are thermonuclear explosions. The entire star blows out. There's nothing left. Type 2s are gravitational collapse.
Are what?
Gravitational collapse. And so a type 1 will start with something about the mass of the sun, close to that. A type 2 will be a much more massive star that collapses gravitationally on itself and often leaves a core behind. So those are the primary distinctions.
Okay, from there I'd like to go to radiations. Given the fact that a supernova created the solar system, we're in what the astronomers have labeled as the local bubble. It's several 100,000 light years. What happens then, since we're therefore shielded so much from radiations as human ever since we came to be, how do we adapt to the fact that we'll run into cosmic radiations and gamma ray bursts when we are able to travel beyond the local bubble in our starship? Started with this 100-year Starship project.
So I mean, I think we are exposed to cosmic radiation already.
Yes.
Sure, but the local bubble keeps out the most harmful types, such as the gamma ray bursts, such as the.
You know, I'm going to suggest that we take this conversation on after the show, so that we can keep things going. I'm going to make you the last question for the night, so let's take the next two. Thank you for that.
Given the, I'm an aging person, and given the literature that we as educated normal people read in the science literature, the New York Times, wherever, the mantra for aging people is exercise, exercise, exercise as the one thing that can increase lifespan and increase quality of life, or at least delay the decay of quality of life. And I'm wondering how exercise fits in with senescence and with inflammation in the model that you're developing.
So there's a very interesting biological phenomenon which is called hormesis. And the idea behind hormesis is a little bit of stress enables you to handle the larger stress better. One of the things exercise does is it actually causes a little bit of inflammation. And the idea is, this is still hypothesis, but the idea is people who exercise moderately, so We're not talking about triathlons, right? Seriously, those people have severe problems later in life.
Now, wait a minute.
Triathlon.
Do you have a question?
But moderate exercise throughout life, one hypothesis is that that low level of inflammation now enables your body to deal much better with disease-causing inflammation caused by other insults.
Thank you. I'll keep on exercising.
Thank you. All right, our final two questions tonight.
So first of all, I wanted to thank all the scientists for sharing your research with us this evening. And I don't know if you were aware of each other's work.
Thank you.
And then secondly, this is for Dr. Campisi and Dr. Ceja-Navarro. We know that insects have proliferated without damaging the earth, and there's how many billions, but if we improve human life, where we proliferate, what would be the impact on the earth and its resources if we were as successful as insects?
Right. Well, for one, I'd recommend we all be short, so we take it. Well, one thing I will say that is interesting that's known by demographers is that when health span and lifespan are extended, birth rates fall. And so I personally am less worried about overpopulation. I think that helping the health of the earth is going to, in the end, benefit us from not overpopulating the earth. And that's a philosophical point of view. As far as insects go, I don't know, but we're extending, the world's record for extending lifespan is in a nematode.
Yeah.
It's C. elegans. We can take that little worm, which lives just a couple of weeks, and extend its lifespan 10-fold. Yeah. I don't think it's had any impact on the soil.
No, well, I mean, I don't think we will reach the point of being as successful as insects are. And even in, no, seriously, I mean, we are humans and we know our limitations, but I mean, despite of that, first of all, if we proliferate, I don't think insects will go away at all. They will still find a way to adapt to us, just as roaches did. You know? So they will be always there. I mean, they were here before. They know how to deal with whatever thing we may want to try. Yeah.
Hey, sir?
This question is for Anne. I realize that at one point science proved that the Earth is not the center of the universe. I'm curious, with the realization of the expanding universe, does this put us once again at the center of the universe? Is it expanding from all points?
Well, so that's the great thing. If we're expanding, everybody's at the center, right?
Right.
We're all moving away from each other. Well, that's good from our radiation standpoint, we decided, right? So there's a whole lot of philosophical questions when you start talking about your universe expanding.
But it's expanding from all points at all times, so every one of us is...
So the way I think about it is ants, we'll come back to the insects. The ants crawling on the balloon. So the balloon, it's a 2D analog of a 3D situation, but the ants, if you blow up the balloon, the ants are all moving apart from each other even if they're not moving. If they're not walking, they're still moving apart from each other.
Right.
And none of them is at the center of the expansion.
But they are expanding. So the ant itself isn't expanding, but
their world is expanding.
Their world is expanding.
So if you like, we can think of ourselves as ants on the balloon. We're all just gradually drifting apart. Some analogies are more palatable than others.
Okay.
But that's how I think of it. They're all their world is expanding. None of them are at the center of it.
Right. Okay.
And on that note,
2D to 3D.
On that note, we will close. You know, thank you, Oakland. You guys have been a great audience. And thank you to our public affairs staff. Particularly, I need to call out, you don't know this person, but you know, there are a lot of people who work behind the scenes. So a woman by the name of Beverly Hill Thomas, who's been working on these for 12 years. This is her final one, so we need to give her a round of applause. You don't know her, but she's great. And thank you to our scientists, of course, for doing such a wonderful job in their presentations. And we'll be in Oakland again. Hope to see you soon.